Dear students, happy to meet you in this online mode of education. I am Dr.S.ANGELINA PARIMALA, Associate Professor and Head from the PG and Research Department of Zoology, A.D.M. College for Women, NAGAPATTINAM. The topic I am going to discuss with you today is about the linkage, which is coming under the paper genetics. Let us see the introductory part. Linkage means what? Linkage and the association word, that is recombination. Linkage and recombination are the phenomena that describe the inheritance of genes. It refers to the tendency of genes that are located close to each other on a chromosome to be inherited together during meiosis cell division. This phenomenon can significantly affect the inheritance patterns of traits, and it is a crucial concept in genetics. In linkage, two or more genes linked together are always inherited together in the same combination for more than 2 generations, whereas in recombination, the genetic material is exchanged between different organisms, which leads to the production of offspring with the combination of traits. The chromosomal theory of inheritance came to the existence long after Mendelian genetics. The lack of advanced technology and physical proof were two reasons for the rejection of the Mendel's concept of genetics. However, later experiments confirmed Mendel's results. Let us see something about the important features. Biologists developed the chromosomal theory of inheritance. The work was further carried forward and proved by T.H. Morgan. This led to the concepts of linkage and recombination of the genes. Thomas Hunt Morgan worked on Drosophila melanogaster, that is fruit flies, to explain how the process of sexual reproduction produced variations. He preferred to work on fruit flies or Drosophila melanogaster for the subsequent reasons. The reasons are it could be cultivated easily on a synthetic medium in the laboratory, and also it exhibits a short lifespan. A single mating could generate multiple progenies. The differentiable male and female sexes is very, very easy, and also hereditary variations are of different types. Morgan carried out a dihybrid cross between white-eyed and yellow-bodied females and red-eyed and brown-bodied males. Self-crossing of F1 generation surprisingly produced and F2 generation without being the ratio of 9:3:3:1, the outcome exhibited a divergence from the Mendel's dihybrid cross in P. Genes are said to be the linked when genes are with different traits are located in similar chromosomes and they are tied with each other. It is a deviation from the Mendelian principle of independent assortment that is appropriate to be applied to the genes that are situated on the different chromosomes. The term genetic regeneration, which is described the non-parental gene combinations in a hybrid, that is in the dihybrid cross. Once the linked genes were identified, the recurrence of linked genes also determined the expression of characteristics in the next generation. A student of Morgan, Stewart, discovered the site of linked genes on the chromosome by estimating their frequency of genetic recombination through the gene mapping process. Let us see something about the basic concepts in linkage, that is chromosomes and genes are segments of DNA located on the chromosomes. Each chromosome carries many genes, which depends on the independent assortment, that is according to Mendel's law of independent assortment. Genes on different chromosomes are inherited independently. When genes on the same chromosome, they may not assort independently. Instead, they can be linked and inherited together. When we see the types of linkages, it can be classified into two types, that is complete and incomplete linkage. When we see in the complete linkage, when two genes are so close together on the chromosome that they are always inherited together. This is rare because crossing over usually occurs. A linkage is said to be complete when two or multiple characters are inherited and normally surface in two or further generations in their parental or original combinations. They are known as complete linkage. These particular genes do not generate combinations that are on non-parental. The genes that exhibit these linkages are located nearby in the same chromosome. For example, genes for long wings and gray body in the male Drosophila. Let us see something about the incomplete linkage. Here it is displayed by genes that generate some portion of non-parental combinations. These genes are situated at the distance on the chromosome, which can be attributed to the occasional or accidental deconstruction of chromosomal segments while crossing over. When genes are close on the same chromosome but can be separated by crossing over. The closer the genes, the less likely they are to be separated. When we see the correlation between linkage and crossing over, here it is a tendency of genes present in the chromosome to stay intact and transfer to the next generation, whereas crossing over is the exchange of chromosomal sections to the disrupt builds links and form new linkages. These linkages generate parental types and increases in age as the linkage increases. Crossing over generates recombinations and it decreases with age, and the occurrence of crossing over between two genes is decreased if they are placed closely, whereas the linkage strength between two genes increases if the chromosome is located in the vicinity. Linkages assist in maintaining a new improved variety. Crossing over, on the other hand, forms a source of variations to produce new varieties. And here in the crossing over, in other terms, it is the exchange of segments absorbed in homologous chromosomes between non-sister chromatids and takes place during the packaging stage of the prophase 1 in the cell division process of meiosis, and also it always takes place within linked genes. The recombination of linked genes that crossing over produces plays a significant role in evolution. When we see the important points on the relationship between linkage and recombination, here Morgan noted that while crossing a set of characters, two genes did not follow Mendel's law as they did not divide as per the law. The likelihood of achieving a parental combination if two genes are situated on the same chromosome was relatively higher in the subsequent generation in comparison to the non-parental combination. The physical connection of genes was referred to as linkage. Additionally, they observed that the likelihood of recombination is reliant on how influential the linkage is. In other terms, we can say that two genes are linked on chromosomes. The genes may or may not be linked tightly. A few genes have powerful linkage, giving a limited chance of recombination, whereas another linkage of genes is closely linked and weakly, providing a great chance of recombination. Let us see something about the recombination. Here it is the process of producing new combinations of alleles by the recombination of DNA molecules. It is also referred to as genetic recombination, as there is an exchange of genetic material, that is DNA, between two different chromosomes or between different regions of the same chromosome. This process is observed in both eukaryotes and prokaryotes. It increases the genetic diversity of sexually reproducing organisms. And also, when we see the types of recombinations here, it can be of two types: homologous recombination and non-homologous recombination. When we see the homologous recombination, this type of recombination occurs between chromosomes of similar sequences and is carried out during meiosis. When we see in the non-homologous recombination, it occurs between chromosomes that are not similar, and also here it is a site-specific recombination. And also it is observed between the very short sequences that usually contain similarities, that is mitotic recombination occurs during interphase. However, this type of recombination is usually harmful and can result in tumors. It increases when the cells are exposed to radiation. In the prokaryotic cells, which undergo recombinations through one of the three processes, that is from conjugation or from transformation or from transduction. When we see the recombination of linked genes, here let us take an example of freckles and red hair to understand the recombination of linked genes. Here it happens in people because of the genes of both present on the same chromosome. During homologous recombinations, the DNA is rarely split between the two genes. Although homologous chromosomes occurs a number of times The two features are inherited together most of the time because of the chances of splitting up of the DNA coding for these two genes are very low. As a result, genes are inherited together most of the time. When we see the recombination frequency, here it is a measure of genetic linkage and is used in the creation of genetic linkage map. Recombination frequency is it is with a single chromosomal crossover. It will take place between two genes during meiosis. Yeah, centimorgan, the unit M is the unit that describes recombination frequency, and in this way we can measure the genetic distance between two loci based on their recombination frequency. This is a good estimate of the real distance. Double crossovers would turn into no recombination. In this case, we cannot tell if crossovers took place. If the loci we are analyzing are very close, a double crossover is very unlikely. And also when distances become higher, the likelihood of a double crossing over increases. As the likelihood of a double crossing over increases, one could systematically underestimate the genetic distance between loci unless one used an appropriate mathematical model. During meiosis, chromosomes are sorted randomly into gametes such that the segregation of alleles of one gene is independent of alleles of another gene. This is stated in Mendel's second law and is known as the law of independent assortment. The law of independent assortment always holds true of genes that are located on different chromosomes. Recombinant gametes are those gametes that differ from both of the haploid gametes that made the original diploid cell. In this example, the recombination frequency is 50% since the two of the four gametes were recombinant gametes. The recombination frequency will be 50% when two genes are located on different chromosomes or when they are widely separated on the same chromosome. This is a consequence of independent assortment. When two genes are close together on the same chromosome, they do not assort independently and are said to be linked, whereas genes located on different chromosomes assort independently and have a combination frequency of 50%. Linked genes have recombination frequency that is less than 50%. The progeny in this case received two dominant alleles linked on the one chromosome. Here, after crossover, some progeny would have received one parental chromosome with a dominant allele of one trait, and also here they are linked to a recessive allele for the second trait, with the opposite being true for the other parental chromosome. This is referred to a repulsion or a trans arrangement. The phenotype here would still be purple and long, but a test cross of this individual with a recessive parent would produce progeny with much greater proportion of the two crossover phenotypes. While such a problem may not seem likely, for example, unfavorable repulsion in linkages do appear when breeding for disease resistance in some crops. The two possible arrangements, cis and trans of alleles in double heterozygotes are referred to as gametic phases, and phasing is the process of determining which of the two is present in a given individual. When two genes are located on the same chromosome, the chance of a crossover producing recombination between the genes is related to the distance between two genes. Thus, the use of recombination frequencies has been used to develop linkage maps or genetic maps. Here, the variation of recombination frequency, when you see it in the chromosome, is an essential process during meiosis. There is a large range of frequency of crossovers across organisms and within the species. Sexually dimorphic rates of recombinations are termed hysteroschemy and are observed more often than a common rate between male and females. In mammals, females often have a higher rate of recombination compared to males. It is theorized that there are unique selections acting on meiosis drivers which influence the difference in rates. The difference in rates may also reflect the vastly different environments and conditions of meiosis in oogenesis and spermatogenesis. Genes affecting recombination frequency, here it is in the mutations in genes that encode proteins involved in the processing of DNA often affect recombination frequency. In bacteriophage, T4 mutations that reduce the expressions of the replicative DNA polymerase increase the recombination several fold. Here the recombination is reduced by mutations in genes that encode proteins with nuclease functions, and also here we can see the DNA binding protein, and also the mutation in the bacteriophage gene also substantially reduces recombinations. And also here we can see these observations in the E. coli. When we see something about the meiosis indicators with very low pedigrees or with very dense genetic markers data, such as from whole genome sequencing, it is possible to precisely locate recombinations. With this type of genetic analysis, a meiosis indicator is assigned to each position of the genome for each meiosis and a pedigree. The indicator indicates which copy of the parental chromosome contributes to the transmitted gamete at the position. For example, if the allele from the first copy of the parental chromosome is transmitted, and it might be assigned to the meiosis, if the allele from the second copy of the parental chromosome is transmitted, and also here it is assigned to the meiosis. The two alleles in the parental chain, one from two grandparents, these indicators are then used to determine identical by distant states of inheritance states, which are in turn used to identify genes responsible for diseases. When we see the common example for linkage is a linkage with Drosophila melanogaster. Here in fruit flies, the genes for body color, gray or black, and wing size are located on the same chromosome. Gray body is dominant over the black, and normal wings are dominant over the vestigial. A cross between a gray-bodied normal winged fly and a black-bodied vestigial winged fly, they produce F1 offsprings that are heterozygous. If these F1 flies are test crossed, the offspring show more parental types than recombinant types. When we see the relationship here, the recombination and crossing over during meiosis, homologous chromosomes can exchange segments. Is the process is called crossing over. Here, the frequency of crossing over between two genes is proportional to their distance apart. This is measured in a map unit and centimorgan units. When we see the linkage map, here the linkage maps of diagrams that show a relative positions of genes on the chromosome based on the frequencies of recombination. Example, if the recombination frequency between gene A and B is 5%, and between gene A and C, here 10%, and between A and B are closer together than A and C. When we see some important points of significance here, the predicting inheritance patterns. Linkage helps to predict the livelihood of inheriting combinations of traits. Genetic counseling is another important phenomena here. Understanding of linkage is crucial in genetic counseling for predicting the inheritance of genetic disorders, and also in the breeding programs in agriculture. Linkage formation helps in breeding plants and animals which desirable traits, and also in gene mapping. Linkage analysis is a fundamental tool for gene mapping, helping scientists identify the locations of genes on the chromosome. And also it is very important in understanding evolutionary aspects. Linkage and recombination rates affect genetic variation and evolution. In example, to human disease genes, here the genes for certain genetic disorders are linked. For instance, in cystic fibrosis, here the CFTR gene on the chromosome 7 is linked with several genetic markers. By studying the inheritance patterns of these markers in families, researchers can narrow down the location of disease genes. When we see the limitations, linkage analysis has a number of methodological and theoretical limitations that can significantly increase type 1 error rate and reduce the power of map in a human quantitative trait. While linkage analysis was successfully used to identify genetic variants that contribute to rare disorders such as Huntington disease, it did not perform that well when applied to more common disorders such as heart disease or different forms of cancers. And explanation for this is that the genetic mechanisms affecting common disorders are different from those causing some rare disorders. When we see the analysis of linkage, it's a genetic method that searches for chromosomal segments that correlate with the ailment phenomenon through families, can be used to create map genes for both binary and quantitative traits. And also this analysis may be either parametric with the relationship between phenotypic and genotypic similarly, or non-parametric. When we see the parametric linkage here, this analysis is a traditional approach where the probability that the gene important for a disease is linked to the genetic marker is studied through the LOD SO, which assesses the probability that a given pedigree where the disease and the markers are conjugating is due to the existence of linkage. And non-parametric linkage analysis in turn studies the probability of an allele being identical by descent with itself. And also linkage map, here it shows this is the first successful gene mapping work that provides important evidence for the chromosome theory of the inheritance. The linkage map shows the relative positions of alleles on the second Drosophila chromosome, the distance between the genes to the percentages of chromosomal crossover even that occur between different alleles. Here the linkage map is again it is having the important position of its known genes of genetic marker relative to each other in terms of recombination frequency, which is rather than a specific physical distance along each chromosome. And this map first developed by Alfred Stewart, a student of Thomas Hunt Morgan. And also when we see the linkage map here, historically the markers originally used were detectable phenotypes, and here we can include enzyme production, eye color, which are derived from coding of the DNA sequences, and also eventually confirmed or assumed non-coding DNA sequences such as microsatellites or those generating restriction fragment length polymorphisms have been used. Linkage maps helps researchers to locate other markers such as other genes by testing for genetic linkage of that is already known to markers. In the early stages of developing a linkage map, the data are used to assemble linkage groups, a set of genes which are known to be linked. As knowledge advances, more markers can be added to the group until the group covers the entire chromosome. For well-studied organisms, the linkage groups correspond to one another with a chromosome. This map is not a physical map or it is a gene map. The occurrence of different genes on the same chromosome, they show non-random assortment at meiosis. This linkage group all the genes located physically on the given chromosome, and also we can see the abstract map of chromosomal loci based on recombinant frequencies, and here the genes on different chromosomes are called independently, giving 1:1:1:1 test cross ratio. Linked genes do not assort independently but tend to stay together in the same combination as they were in the parents. When we see the gene mapping, it refers to the process of locating genes within the genome. These regions of genome that directly encode proteins, and the protein coding genes are usually great interest to scientists. Therefore, it is often a priority to locate each gene in the genome. Nowadays, the mapping procedure typically involves genome sequencing and analyzing the results which we are getting in the digital methods that would lead to access for desired genes. Therefore, more gene mapping projects begin in gene sequencing. And also, when we see the simple definition here, it refers to the techniques used to identify a gene's location and the distance between genes. The distances between various sites inside the gene and also can be described through the gene mapping. And also, placing several molecular markers at specific locations on the genome is a fundamental element of all genome mapping. There are many types of molecular markers. When creating gene map, and here it can be observed as a particular class of genetic markers mapped similarly to other markers. When we see the types of gene mapping here, we can see the map which are having main characters with gene mapping, physical map and physical map and or main categories which are falls into the genetic markers. While physical maps involve actual physical distance often measured in number of base pairs, distance of genetic maps are based on genetic linkage information. There are many gene mapping methods including comparative physical and genetic linkage mapping. However, physical and genetic linkage mapping are more common. And when we see here in the genetic linkage mapping, it shows the location of each gene on the chromosome and their relative distance from each other. And also initially these maps were created by tracing the inheritance of several features like eye color and hair color, and also blood, saliva or tissue samples from both affected and unaffected family members are used to begin a genetic map. Saliva is the most often used sample in genetic mapping, particularly personal genetic studies. And also it is possible by crossing over a regular biological occurrence during meiosis, and also chromosomes line up in pairs in the center of the cell during the first phase of meiosis, where they can often stick to one another and exchange similar fragments of themselves. And also in the gene mapping, here it makes it possible to determine which gene is present on each chromosome and where it is located within the specific chromosome. And also it is based on the distance between two genes. Mapping can also determine which gene is more likely to undergo recombination. When we see the physical mapping here, it provides the actual DNA based on distance between landmarks, and it is one of the gene mapping approach that has a high degree of accuracy in determining the DNA path. And also here we can see the radiation hybrid mapping, sequence mapping, and cytogenic mapping are the techniques which are used to produce a physical map. And it is also assembles larger DNA sections using DNA markers and DNA fragments. And also researchers can identify the locations of the DNA bases from the overlapping sections of the fragments. And also here it is a common approach used in genome sequencing to acquire an entire genome sequence and determine whether there is any correlation between the specific DNA sequence and phenotypic features. When we see the genetic mapping here, it is used to measure distance between genetic markers and also random fragment length polymorphism. Here it helps calculate the distance between two markers, and also current gene mapping analysis targeting single gene disorders is used to repeat the polymorphism. And here the genome-wide assertion studies the connection between the traits and markers like SNPs and microsatellites by treating the population as a single family. Here this method is used to map the gene functions of common disorders. Let us see something about the theory of linkage. Here Morgan along with Castle formulated the chromosomal theory of linkage, which is as follows, that is the genes which show the phenomenon of linkage are situated in same chromosomes, and these linked genes usually remain bounded by the chromosomal material so that they cannot be separated during the process of inheritance. The distance between the linked genes determine the strength of linkage. The closely located genes show strong linkage than the widely located genes which show the weak linkage. The genes are arranged in linear fashion in the chromosomes. When we see other characters, the genes for different characters may be either situated in the same chromosome or in the different chromosome. When the genes are situated in different chromosomes, the characters they control appear in the next generation together or apart depending on the chance alone. They assort independently according to mental laws of independent assortment. And here we can see in this type of coexistence of two or more genes in the same chromosome, and here it is specified as linkage. When we see here the factors that shows linkage, disequilibrium or the rate of genetic recombination, mutation rate, genetic drift, system of mating, and population structure. Let us conclude it. Linkage is a fundamental concept in genetics that describes how genes on the same chromosome are linked, inherited together. By studying linkage and recombination, scientists can better understand genetic inheritance, assist in breeding programs, predict the genetic disorders, and map the locations of genes on chromosomes. Thank you.